Welcome everyone again to another episode of the Idea Me show that profiles the humans behind the really big ideas that are shaping our world, inspiring the future and future creators. And for all those that like great science and great stories, I'm Ira Pastor, your health, aging, and longevity ambassador along for this journey today. Today we are going to take a journey into the area of our mitochondria. And for those that may or may not be familiar with mitochondria, mitochondria are these double membrane bound organelles. They are found in most eukaryotic organisms, and they are basically responsible for generating most of our cell supply of ATP, otherwise known as adenosine triphosphate, used as a source of chemical energy. Hence, the mitochondria has been given the term for many years as the powerhouse of our cells. Ultimately, damage and dysfunction to our mitochondria is an extremely important factor in a wide range of human diseases, both very common and rare, due to the influence of cell metabolism and many processes. Mitochondrial disorders can present themselves as neurological disorders, but also manifest in a variety of other conditions, myopathies, diabetes, various endocrinopathies, many other systemic disorders. And our mitochondrial genome, which we've talked a little about in the past, represents this genetic complement of somewhere around 16,000 or so base pairs representing 37 genes that primarily encode for proteins that are responsible or component of the oxidative phosphorylation system, and that enzyme of complexes. Mitochondrial dysfunction can be driven, of course, by damage to our mitochondrial genes, but at the same time, there are other diseases where it's genes within the nucleus that are responsible for mitochondrial dysfunction. That can be the case of certain rare diseases, featured ataxia, paraplegia, Wilson's disease, and so forth. Today on the show, I'm very pleased Dr. Magomed Kaitakov is joining us. He is a research assistant professor in the Department of Internal Medicine at University of Arkansas for Medical Sciences. Dr. Kaitakov has both an MD from First Medical Institute in Moscow, Russia, and a PhD in molecular biology from University of Victoria in British Columbia. He is also the author of The Pessimistic Guide to Anti-Aging Research, what we'll get into a little bit further on the show. His interests are a wide range of biologies involved in aging and degenerative pathologies. He's interested in identification, ultimate correction of various so-called biological design flaws that may limit mammalian lifespan in certain organisms. Dr. Kaitakov, thank you for taking the time to come on the show today.
Pleasure is all mine.
No, it's great. It's great having you. We typically start off the show by giving our guests the floor just to talk about themselves for a little while. If you can take us sort of back in time into your journey, sort of how you got started, your background, where you grew up, how you developed an interest in science, molecular biology, medicine, and ultimately how longevity and aging has become a very important interest and target of yours.
Well, gerontology field is filled with bright personalities. I'd rather cut down on introduction, but yes, I was interested in aging for a long, long time, and my most valued mentor, Robert Schmuckler-Rees, also at the University of Arkansas for Medical Sciences, he was my mentor, and I really do appreciate his mentorship because he's an amazing guy. And if you take a dozen random gerontologists, he knows more than the whole dozen. So anyways, my interest in human aging is not limited to mitochondria.
Sure.
As a matter of fact, I would prefer to speak or to talk about several independent processes driving aging, independent clocks, which by definition implies that any kind of intervention aimed at a single mechanism, single independent mechanism,
sure,
is essentially doomed to fail. Especially considering that, judging from experimental data, these mechanisms are more or less synchronized. So technically speaking, removing one will not change dramatically anything. And that's about it. As far as my biography is concerned, it spans two continents, three large and beautiful countries. First Russia, then Canada, then beautiful United States of America. And it's not an idle flattery. It just expands your horizons, and you understand that world is enormously big and enormously diverse and enormously beautiful.
Absolutely. Absolutely. From Russia to Canada to beautiful rural Arkansas.
Absolutely.
I see the pattern. I definitely, I appreciate the comment and completely agree with you 100% about the unique systems nature of this aging thing. I just, with some of the work you've been doing, some of sort of the plans or some of the research in terms of mitochondria, I thought we'd start there, but we can definitely, I have a lot of time. We can go into many other areas. Well, why don't we start out there though? Because it's, it's an area that we, it gets talked about a lot as an interesting target, of course, because of what mitochondria do. Take us just on a little journey there of mitochondria in general, because obviously there are people going to be watching the show that know quite a bit about it, many that won't. So why don't we start there, and then if you could sort of give a general overview. Sure. Going to go into a lecture on mitochondria, but if you want to talk about mitochondria, talk about the relevance in aging, and then in my next series of questions, I'll go into some of the plans you have specifically for investigating areas like the deletions and so forth.
Well, mitochondria, despite the fact that it's quite popular in contemporary research, is an overlooked item.
Sure.
Just Absolutely. To give you a small few but very impressive numbers. So we have, you mentioned this mitochondrial genome, mm-hmm. just about 16.5 kilobases, mm-hmm. which is coding for 13 proteins absolutely essential for oxidative phosphorylation, mm-hmm. and production of ATPs. Okay, so, and there is a reason why this mitochondrial DNA stayed in there. Because originally mitochondrial DNA was much larger, but most of it was gradually transferred to the safer environment of the nuclear DNA. And the question arises why that little portion of of DNA remained in mitochondria.
Okay.
And there are quite specific logistical reasons. Nature could not handle those reasons, but maybe mitosense will, I don't know. Although I have certain doubts about that. Simply because that proteins coded by mitochondrial DNA are extremely hydrophobic. Right. And as soon as you move them out to nuclear DNA, you have a logistical problems of delivery inside of mitochondria.
Sure.
And that appears to be unsurmountable for the nature. Maybe using modern technologies will be able to overcome this problem. But I do have some reservations about the feasibility of this project. Mm-hmm. I would imagine that one or two may be somehow handled, and DNA for coding for these proteins can be moved to nuclear DNA. But overall problem, I do not think will be solved using this methodology.
Okay.
Okay, and then another number. In the course of life, we have accumulation of mutations in mitochondria. Mm-hmm. Those mutations tend to clonally expand occasionally.
Sure.
And occupy a large fraction of population of mitochondrial DNAs in the cell. So when the percentage of mutated mitochondrial DNA reaches more than 70%, mm-hmm. sometimes 80%, cell is unable to process oxygen and therefore becomes respiration deficient. And for your information, in our twilight years, 30% of our brain cells become respiration deficient because of this process. Similar numbers were observed in skeletal muscles. Similar numbers were observed in epithelium of intestines. So as you can see, 30% is not a trivial number.
Yeah.
It's an enormous number. It's functionally crippling number. And that problem was not addressed. Well, MitoSense project does address it, but in my opinion, with very slim chances for success. So there are different ways to deal with this problem. And this is a representation of one of the independent clocks that determine our lifespan, or at least our functionality and our cognitive abilities in our twilight years. So here we have a choice. No, we don't have a choice yet.
Not yet, hopefully, but surprise us.
I'm sorry. Yes, but I'm constantly vaguely amused when I look at the faces of yet another messiah in gerontology, and his eyes are just emanating conviction and holy zeal, but it is unfounded, honestly. Unless we approach the problem as a complex, extremely complex phenomenon that has many different independent inputs, we will never solve the problem.
Sure. You, a couple of years ago, you have a paper out there. It's entitled Species-Specific Lifespan, Can It Be a Lottery Based on the Mode of Mitochondrial DNA Replication? And you basically talk about these large deletions that lead to many of these, sort of the downward spiral deterioration in terms of mitochondria, the senescence, and then ultimately cell death. And you've done some comparison research in shorter-lived and longer-lived species that have potential to keep these deletions at bay, or they occur at a slower rate and so forth. Can you talk a little bit about that and some of what you've discovered over the years, and then once again coming in now from your thinking about maybe some easier ways to approach this problem earlier on before we get into the sort of the point of no return and a more downward spiral. Tell us a little about what your thinking is there, what you've learned, where you think we potentially are going with let's say I'll use the term more translatable possibility.
Well, first of all, whatever I write is at least 20% an irresponsible babbling. It's conjecture, which has not been experimentally proven. But what is not conjecture, that the vast majority of clonally expanded mutations found in neurons and myocytes are large deletions. These large deletions are flanked by direct repeats, so it's a homology-driven process. And it's partially linked to replication, to the mode, very specific mode of replication of mitochondrial DNA, and partially linked to repair of DNA when there are strand breaks. So, evaluating the situation from that angle, we have a number of options. We can edit our mitochondrial DNA in such a way as to reduce number of direct repeats, for one thing. We can try to modify the mode of replication of mitochondrial DNA and make it more similar to nuclear DNA. In this way, to prevent major mechanism that is responsible for formation of these deletions. Also, there is another rather unexpected possibility. When we were analyzing telomerase, we did the analysis of docking to the origin of replication for the light strand, and it turns out that telomerase really binds the origin of replication in mitochondrial DNA. And this way, it affects the process, the mode of replication.
Interesting.
So it's quite possible that the effect of telomerase on longevity is not really linked to lengthening telomeres, but rather it is linked to its mitochondrial action through modifying the process of mitochondrial DNA replication. So it's becoming an interesting puzzle. it's becoming a truly interesting puzzle, because if you're talking about telomeres, there are so many controversies.
Sure.
Because in original studies, when they overexpressed telomerase, the animals were dying from excessive tumorigenesis.
Right.
Then somehow, miraculously, 5 to 6 years later, they stopped dying from excessive tumorigenesis. I don't know how to resolve this issue, but considering that the processes are more or less synchronized, so we have an accumulation of mutations in nuclear DNA.
Mm-hmm.
And in this context, shortening of telomeres prevents those potentially cancerous cells from acquiring the tools to continue their existence.
Mm-hmm.
So these processes are interlinked. I think that expectation that if we extend our telomeres and we'll give our healthy cells a chance to live longer is somewhat exaggerated, because it gives a chance to bad cells to continue as well.
Sure.
And the older you are, the greater this chance becomes. So telomere extension is not necessarily a conceptually clean way to extend human lifespan.
Right. That was that's a very sort of elegant example of, as you were saying, the various clocks, there's interconnection. Talk about other areas now. If we move beyond the mitochondria,
sure,
beyond the telomeres, take us on take us on a journey to more of these clocks, because I'm obviously this area has been a battleground over the years between these damaged folks on one hand, the program people on the other. No one ever wins that fight, but it goes on. But clearly, with all this data coming out about epigenetic clocks and other forms of programming, there's a lot, as you were saying earlier, there's a lot of connections in this system, some which we're just learning about. Take us on some other journeys that you were.
I want our listeners to appreciate majestic complexity of life.
Please do.
And as far as epigenetic goes, of course, the concept is beautiful in many ways, especially with the Yamanaka transcription factors. Yes, scientists were capable to rejuvenate even cells from centenarians. Mm-hmm. But all this is done in tissue culture. Right. So, and now consider the heterogeneity of tissues in the living organism, in such complex organism as ours.
Absolutely.
So, true, we can rewind the epigenetic clock back all the way to undifferentiated state. And that creates a logical, using formal logic potential to do this process just partially and eliminate aging-related changes in epigenome. So far, so good. But first consideration, according to modern school of thought, the majority of cancers come from stem cells. And we're returning cells, our old cells, somatic cells, to stem-like condition. Mm-hmm. All compromised cells, again, will have the possibility to reveal themselves in the form of cancer or other conditions. And the problem with epigenetic rewinding of clock is because it's based on transcription factors, it's extremely difficult to regulate this process within the therapeutic range. I don't even see how it can be possibly done, but never mind me, because I'm not an expert in this area. Maybe someone else will be able to do that. But to me, it seems still that it's a very immature field. And also it will be always fraught with danger. It's similar to the, for example, this fugu fish, which is poisonous. And in Japanese restaurants, only trained chefs are preparing this dish. Yet, from time to time, the patrons get poisoned.
Mm-hmm.
And this analogy applies to epigenetic way of treating aging processes. There is always a possibility, a very real one, that you get your poisoned fish, despite the qualification of the chef. So all cells in the organism, in the process of aging, accumulate their own unique collection of mutations in nuclear DNA.
Sure.
According to some estimates, it's up to 5,000. According to other corrected estimates, it's about 1,000 mutations, maybe slightly more, per cell.
Right.
And our coding sequences occupy just about 1% of our genome.
Mm-hmm.
So let's say 10 mutations in where it matters, in coding sequences of genes. Let's also consider that our genes are protected in many ways, and only about a third of mutations are potentially harmful. So let's say 3 mutations per cell in the course of life
Mm-hmm.
will be potentially dangerous. But the collection of these cells is unique, of these mutations is unique for every single cell. So they're not identical anymore.
Yep.
And when you think of it, if you think of your body or my body in my age, so it's like a team of sled dogs. When they're young, all their bodies point to the same vector, have the same vector and point in the same direction. In our older bodies, yes, they generally follow the same direction, but they fluctuate a little bit. Their vectors, individual vectors, may point slightly off. And that's what is happening to our organism. It's
Mm-hmm.
not critical as far as nuclear mutations are concerned. It's not critical at all, but it creates a huge problem when you attempt to rejuvenate using epigenetic clocks.
Mm-hmm.
So epigenetic approach to curing aging may make sense if we address the problem of mutations as well.
Sure.
So nothing can be touched in itself. You need to touch it everywhere.
Mm-hmm. Mm-hmm.
And that's my problem. And that's your problem. And that's everybody's problem. Unfortunately, gerontology right now is mostly based on unshakable faith in a certain specific intervention.
Sure.
And it's going to work.
Yeah, it's a model that unfortunately takes its cues from, I point out the last 100 years or so of the pharma industry's sort of single magic bullet thinking, which we definitely will have to go beyond, in my opinion, to have an impact here. There may be beneficial pharmacotherapeutic interventions that come out of such targeting, but when it comes to the system's nature, as you elegantly talked about, we have to think quite differently about how we approach these things. And in that, I'd like to sort of segue to your book and your series of videos. You wrote the Pessimistic Guide to Aging Research. You put out a series of videos based on the different things you talk about. I don't really view it as pessimistic. I think you like some other folks, you're much more pessimistic from the translational perspective. I think you think there's a lot of good stuff out there, a lot of great possibilities. But as we've discussed, there's really going to be you have to be unique translational thinking and unique systems thinking in creating something that looks probably a lot different than the little pill that we get at our local pharmacy. There are going to be interventions that are different than we think, more complex. Talk about why you wrote the book. Talk about why you make the videos. Obviously, there are components of the longevity community, the advocacy community, that hate any pessimism. You say one negative thing anywhere, that you get excommunicated.
Yeah.
But I've had people on this show that, pretty smart people from the pharma industry that have been around a long time, that say, look, they believe this stuff can get done, but you're not talking about a clinical trial of 5 people for a month. I mean, this is aging. This is, we have to think much bigger. We have to think differently. Talk about writing the book. Talk about the any negative feedback you've gotten from other luminaries, positive feedback, why you did it in the first place. Talk a little bit about all that.
No, I recognize intelligence of leading people in the field. I like Aubrey de Grey. I like Kurzweil. I like all of them. They're highly intelligent, highly professional people.
Sure.
We probably do not share points here and there regarding how aging goes and how to deal with it.
Sure.
Generally speaking, my impression is that everybody honestly is trying to do something about it.
I quite agree.
And I desire immortality as much as the next guy. But to me, it's far more important to address the problem in a realistic way.
Okay.
And I'd say that the state of gerontology now is not conducive to finding solutions. For example, we use, by necessity, various animal models.
Of course.
So C. elegans lives 3 weeks, mouse lives 2 or 3 years, and so on and so forth. But, for example, C. elegans, when you apply antioxidants, you increase lifespan dramatically. Lifespan of C. elegans does not depend on mutations in mitochondria, mutations per se, in nuclear DNA, and telomeres.
Sure.
And even epigenetic control is uncertain at this point. So, as a model of human aging, C. elegans contributes practically nothing. Yes, of course, we can study fundamental processes, just how they work, but not details pertaining to the human aging.
Sure.
Let's take mice. Yes, telomeres play a certain role. The mutation rate is considerably higher in mice. So it's more or less similar, and epigenetics, it works. But mitochondrial DNA mutations have no impact on mouse. And we're not even talking about differences in physiology and so on and so forth.
Right.
And it's not surprising at all. And another point, as a justification for using animal models for research on human condition, we emphasize similarities. And somehow we forget to emphasize on crucial differences. And there are many examples. The success rate of clinical studies is barely 8%. And I'm not talking about mirroring effects on experimental animals. No, we're talking about at least some confirmation of experimental studies on animals. And it's a dismal percentage.
Sure.
That indicates that we're doing something terribly wrong.
Absolutely.
So our strategy of approaching human conditions through animal experimentation is probably the only one at present. But it's not good. So we need to think of something else. And you are absolutely right. For example, there was I think it was TNF inhibitor or something like that, that was confirmed on different studies, including monkeys. And then there was the pilot clinical study with 6 or 8 patients. And a few seconds after injection of this medication, people started dying.
Yep.
Which highlights the importance of minuscule differences, even in physiology. So I would evaluate our chances to get anything from animal studies as close to zero, technically speaking. So we need to change our method of analyzing aging in human beings and finding a cure for aging in human beings. And I don't know how to approach this. I just know that this problem exists.
Yeah, no, I totally agree with you. You say you don't know what to do, but I think you might have some ideas. If I'm going to propose this question, and let's have a little fun with this. If the skies opened up and, $100 billion fell out of it tomorrow into your bank account, it comes with the caveat you can't spend it on yachts and other things. What would you? I believe that I love talking to folks like you. You're extremely smart in terms of what's going on. I'm sure you have some ideas of what you would build, how you would reconstruct the system that exists now. I don't know if that's using artificial intelligence or virtual clinical trials or creating unique monkey human chimeras to do better testing. Who knows? But if you could use that money to build something to do it better, what are some of the things you do? I'm sure we all have our fantasy ideas of stuff that
Okay.
we would like to do, but we probably can't do it in 2020. But if that money ended up on your table, say, all right, Dr. Kadikoff, we're going to give you this opportunity to sort of rewrite it all. The pharma industry, we'll put them over there, and we'll try a different set of approaches. What do you think can get us there? What, as bleeding edge as you want to go with this? Take it out a little bit.
Apart from fleeing to Siberia and building a palace.
Yeah, you can't go there, and you can't go to North Korea.
It's okay. There are many places a person can hide, but scientifically, I would limit myself only to human cells, human organoids. You see, the sequence can be shortened and made more efficient this way, especially considering that there are so many potential volunteers around here. You cannot prove or disprove that your remedy works unless it is tested on human beings. So I would evaluate the effect on cells, on organoids, and after getting satisfactory results, I would run a very limited toxicity studies on real human beings. And having proven that it's not toxic, I would go for long-term, let's say 5 years at least, study, which implies that I need to have in my hands a panel of reliable indices indicating that aging is reversed, delayed. Otherwise, it doesn't make any sense. I'm not interested in epigenetic rejuvenation alone, because alone it doesn't tell me anything. I will not be able to do anything as far as aging people are concerned with mutations. I simply can't. You cannot remove mutational burden once it's already accumulated.
Sure.
So, man, it's a dilemma. I think the world of rejuvenation will be divided sharply in two parts.
Okay.
The first part is intervention made on the level of zygote.
Okay.
So those will be genetically modified human beings. That's the only way you can involve all cells in your organism and delay the process one on another.
So in essence, sort of CRISPRing in various fail-safes.
But it needs to be done. It will take several decades, perhaps.
Sure.
And a lot of experimental work, but it's possible technically to create a superior human being in terms of lifespan.
Sure.
The rest, of us, like you and I, will have to deal with the patchwork and the eternal hope that someone will help. But technically speaking, it's not possible. Biologically speaking, it's not possible to get rid of all the damage we already accumulated. There is another option, though, because all other industries adopted it already. Why not gerontology? Instead of, you know, that modern world can be characterized by the loss of craftsmanship. We don't have true masters that can go inside of some complicated device, find the cause, fix it, and put it back. The modern strategy is you take this unit out and put a brand new one because it's sensible. So stem cell therapy, organ regeneration, they follow the same philosophy. We don't need to know what is happening. We don't need to know the aging process in every intricate detail. We simply take a faulty unit and put a new one. So the stem cell therapy is using this philosophy. But we as adults who are hopelessly aged already need to take it a little bit further.
Yep.
And it goes beyond the realm of biology at this point. So why don't we take as a unit our entire body and replace it by clone? And it creates a problem of transfer of the memories of personality. And that this is not a biological problem, at least not in its entirety. We don't know how memory operates.
Not at all.
We don't know how it is recorded, whether it can be decoded or reproduced. But it's a feasible possibility for adults like you and me, for all living, for that matter. Although I cannot imagine that it's going to be a cheap solution. So it's going to be available to the happy few. So we have on two extremes of this scale, we have two solutions. Modify on the level of zygote. And for those who are too late for that, to replace the worn out unit and transfer the memory from one clone to another, which would guarantee a practical immortality in a sense. But it's so far-fetched. It's just, again, irresponsible conversation. But these are logical possibilities.
Okay, the discussions that I maintain that these are discussions that that need to be had, even if they are. I mean, yeah, no matter how far-fetched some of the ideas are, we've had sort of nearer-term stuff on, people talking on this show. We've had people talking about stuff that's 500 years out, but ultimately stuff that's 500 years out, it's probably good to start talking about some aspect of it today.
But there's a certain beauty in it.
Sure.
No, in a sense that even at extreme age, our somatic cells did not accumulate that many mutations.
Right.
So it's technically possible to devise a selection process that will yield a perfect clone, almost identical to you, and perfectly sound, functionally sound, and genetically sound cell.
Right.
So in effect, we'll rejuvenate this cell, and all components are present. Epigenetic, rejuvenation, everything, because you're starting up from a zygote again, or its imitation. And you have brand spanking new, beautiful body of yours. And the only problem is to fill its head with the content that you accumulated over the years.
Yeah. That's a tough one, but I have fingers crossed someone will figure that one out too.
You will need
at some point.
You will need to invite Elon Musk for this purpose, because he's got the money and the energy.
Yeah. Speaking about that, I mean, you spoke earlier on about Dr. Reese and sort of important influencers in your career today. Are there other people that you have influenced you, maybe beyond sort of the biological sciences? Other people that that could also be in biology. People that you might want to give a shout out to that have sort of kept you on this path. That if it wasn't for them, you would be doing something entirely different with your time, or anyone else you want to single out and just give a shout out to?
Sure. Just I was lucky. I don't know how you feel about your life and your mentors, but as far as science goes, I was extremely lucky because all my mentors were old school scientists,
Mm-hmm.
encyclopedically knowledgeable, just and who retained curiosity to the very end of their lives. And I remember when I was a graduate student a long time ago back in Russia, my first, and my boss, Professor Spirichev, who itself, who himself had a very peculiar career because he was trained as a diplomat and ended up as a professor in biochemistry.
Mm-hmm.
So, and the atmosphere in our lab was such that I could run into the lab, into his office, open it with my foot, and say, I have an idea. And he would immediately drop everything he was doing and quietly listen to me. And then next morning, I would find a pile of articles related to this topic. This man had no prejudices, no reservations. Uh, well, I wish I could be like him. I mean, amazing people, just amazing. And in Victoria, Barry Wayne Glickman, professor, he's retired now,
Mm-hmm.
somewhere at sea, I suppose. Same type of old school personality.
Mm-hmm.
No prejudice, no mental blocks, immense curiosity. And Bob Reese,
Sure.
I can never attain his level of knowledge, and I will not even try, but he is an enormously talented man. So I'm grateful to all these people. As simple
That's
as that.
nice.
What about you? Did you have anybody like that in your life?
I'm I'm not a scientist. I was trained as a pharmacist that went to business school and got my learning, my teaching from hanging out in the pharmaceutical industry. And I was fortunate enough to have sort of uh, been raised through the business development, sort of corporate development path, where I got to be at the epicenter of sort of everybody's stuff. So I was inside big pharma. I negotiated against smaller biotech, and in biotech, I negotiated against big pharma, and then kept the cardiology team from fighting with the neurology team and the oncology team and all that. So I got a taste for a bit of everything. But yeah, I met some pretty impressive folks along the way, and coming back to our conversation though, one of the things that I appreciate about it, sometimes you get, and I'm not disparaging scientists by any means, but sometimes people, ask these questions about, whether it's an advocacy group or someone starting a new company, what's the most important thing? And people say, well, we need the best scientist here um, to guide us on this particular path. And I say, well, yeah, that's all well and good, but I met a lot of really, really good non-scientists that, spent time crafting this sort of existing industry that is the pharma industry. They may not have been scientifically that savvy, but they were translationally savvy. Something we talked about earlier. They knew how to get products, goods, services to doctors,
Mhm.
patients, and payers. And that's one thing in my personal, sorry, I'm going off here. You're interviewing me, but I believe we need a little more of that angle represented as well, because science is wonderful, but really having those minds and business developments, people that have been out there, that have interfaced in the commercial world, that know what ultimately that prescriber, that patient, and that payer want, is a
The only
very
problem,
important stuff.
the only problem is they speak different languages, unfortunately.
No, yeah, but on the positive, on the positive side, they at least have, and as imperfect as we both agree it is, they have created a system to get, albeit imperfect, materials, sort of this first generation, sort of deal with pharmacotherapy and therapeutic interventions for more symptomatic stuff. If we could change that and utilize that same skill set, I think it'll be useful for some of the things that are coming down the pipe. And so we don't make, as you point out, we've got people that old school folks that have sort of been there. They know the history of the science. One of the big problems that I also see, I don't know if you see this that much, is sort of things that were discovered 3 decades ago that we just don't forget about and we rediscover. There's not enough people, I think, at the table that know how much has already been done in this space. And we too often, I think, try to reinvent the wheel, but that's just that's my 2 cents. But anyway, but this show is about you and getting your voice heard. Any final messages you want to get out there about your current research? I know you're doing some crowdfunding for one of your projects. I'm going to give you the floor again. Take it away. Anything else you want to wrap up with? Love to hear about it.
Biology can be very rewarding, intellectually.
Sure.
So I spend my life thinking about biological problems and petrified in the face of majesty of life and deeply interested. And I think aging is to some degree solvable.
Mm-hmm.
My problem is, however, that maybe it's a lack of developed channels between gerontologists and the public and investors, perhaps.
Definitely.
So I do have this ability to be entrepreneurial like David Sinclair or Aubrey de Grey. What dismays me in a way that when you're talking about cancer, and I mentioned it in my last update to my crowdfunding effort, if I come to you and say you have cancer, your immediate reaction, God forbid, of course, your immediate reaction would be despair. You will be running around like headless chicken, trying to find a good doctor, trying to find any kind of hope.
Mm-hmm.
But your reaction will be visceral and immediate.
Sure.
Because you need to deal with this crisis. At the same time, if I tell you that you are aging, you see your heart rate didn't even go up a bit.
Yep.
Nothing changed. Because there is a sense of finality, common fate, if you will, inevitability. Sure. You can do nothing about. And that's why investing into gerontology is so much lower than investing in cancer or cardiovascular research. Because with all these separate diseases, you have immediate visceral response of people. And for some reason, you don't get this visceral response from people when you're talking about aging.
Yep.
So our main purpose is to destroy this perception of inevitability and that nothing can be done about it. Something can be done about it, at least to the tune of 10, 20, 25 years, which in itself is a very great achievement.
Yep.
That said, I want to invite everybody to try to destroy this image of aging as something inevitable, common fate.
I completely agree with you, and that's as we say, moving the human story forward to get everybody thinking that way. I think that's a wonderful analogy. I never had anyone put it that way, but it makes complete sense when I'm out there talking about it, or a lot of these other people that I have on the show. It's a very elegant way of looking at it, and definitely very much appreciating your insights on that matter. For everyone that's going to be listening to this on the podcast networks, or watching on the YouTube channel and the blog, we've been spending time today with Dr. Magomed Khaidakov, Assistant Research Professor, Department of Internal Medicine, University of Arkansas for Medical Sciences, also the author of Pessimistic Guide to Anti-Aging Research. Check out his work, check out his links, check out some of the projects he doesn't want to talk about because he's so modest, but doing really amazing things in this space, looking at things from a very, a disciplined perspective. And that's not a pessimist. But he's a realist that there's real issues here that we got to solve alongside of the science. And I think that's an extremely important message as all of us are out there talking to the public, talking to investors, talking to government figures and stuff, because there's a lot of people that are going to have to come to the table to make this 20, 25, 30 year perspective a reality. And it's just another very important way to look at things, a very important thought leader to have on the show. And Michael, I really appreciate the time today that you spent with us, and everything that you're doing in your own way, whether it's in Russia or Canada or the U.S. to, as we say on the show, to move the human story, or Siberia, or wherever else your travels may take you. But really, thanks for the time today. It was an honor having you today.
It's been a pleasure.